coordinator is not available, so at least she will be able to see your demo later. Thank you so
Okay,
much,
I
Miss.
already.
So if you're ready, Miss, let's start.
Okay. Um, okay, hello, good morning. Today I will explain about the structure and function of the DNA. Then, what is DNA? We know that inside every cell in our body, there is a nucleus that contains our genetic materials. That genetic materials is deoxyribonucleic acid, and we call it DNA for short. And the DNA is control many of things about us. So this is why the DNA is most of important molecule for life, because it control all your trait. Your cell cannot function without it, and how tall you are, how color your eyes are, and how color your hair is, and then even if you are at risk for a certain disease like a heart disease and then the cancer disease, some of that can be found in the DNA. And then here are some facts of the DNA. First, almost every cell in your body contain about 3 billion DNA. And then the DNA is divided into two sets of 23 chromosomes. And you get one set of chromosome for your mother and from your father. And every time your cell divides, the information is copied and divided into the new cell. And every time those chromosomes are passed from mother or father, then they are recombined to make a unique chromosome and will become your chromosomes. In another word, we can say the DNA is a group of molecules that is responsible for carrying and transmitting the hereditary materials or the genetic instruction from parent to offspring. Okay. On the other hand, the DNA is a helical double-stranded molecule that occurs bound to protein chromosome in the nucleus, and as unbound circular DNA in the cytosol and prokaryotes, and in the mitochondria and chloroplast of eukaryotic cell. And it is an organic compound that has a unique molecular structure. Now we can see this image. We have a cell. Inside of cell, we have a nucleus. And then inside the nucleus, we have a chromosome. And then in the chromosome, it contains the DNA. On the other word, we can say that the DNA coil up, forming the chromosome. And each chromosome has a single molecule of the DNA in it. Okay, this is the DNA type. First, we have an A DNA, B DNA, and Z DNA. And this is the structure of the A DNA, B DNA, and Z DNA. For B DNA is the most common DNA conformation and is a right-handed helix. Okay, let's move to the structure of the DNA. Here, structurally, the DNA contains from the nucleotide. It contains a sugar, phosphate, and then base. Here, we know that the nucleotide is made up from the phosphate, sugar, and base, where the sugar is one which forms the backbone of the DNA molecules that we call deoxyribose. And then for the base here, it determines the genetic code of the DNA instruction. Okay, let's see this double helix shape of the DNA. We can see here that nucleotide hooked together to make the sugar phosphate backbone of every strand of DNA. And then bases pair up with the opposite matching bases to other strand of the DNA, like this image. And we can see here there are 4 kinds of base pairs made up: adenine, thymine, guanine, and cytosine, where the A always pairs with T and G always pair with C. And then from this image, we know that the 2 strands of the DNA are linked together by weak hydrogen bonds. Then we should remember the Chargaff rule. Erwin Chargaff, a biochemist, discovered that the number of nitrogenous bases in the DNA was present in equal quantities. The amount of A is equal to T, whereas the amount of C is equal to G, where A and G are purines, C and T are pyrimidines. And in other words, the DNA of any cell from any organism should have a 1:1:1 ratio of purine and pyrimidine bases. Now we see this image. We have a building block of DNA strand, double strand DNA, and then double helix DNA. What do you get from this image? Okay, now we focus to this double strand, and we can also see this DNA double helix. For this strand, we can see that the 2 DNA strands here are called polynucleotide, where they are made from the simple monomer called a nucleotide, this one. And each strand here has a 5-accent end with phosphate and 3-accent end with hydroxyl groups. And the strands here are anti-parallel, meaning that one strand runs in 5-accent to 3-accent direction and 3-accent to 5-accent direction. It means that the deoxyribonucleotides are linked together by 3-accent to 5-accent phosphodiesterase bond. Here's okay, we can see this image. First image for A is a space-filling model of 1.5 turns of the DNA double helix. We can see here the center to the center distance between adjacent molecule is about 3.4 nanometers, and the diameter here is about 2 nanometers. From this image, we also know that the coiling of the 2 strands around each other create 2 grooves. We call it a minor groove and major groove. For minors is for the smaller groove, and this one for the wider groove. And then the minor groove actually occurs when the backbone are far apart, and then for the minor groove is occurs when the backbone are far apart, and then for minor groove is occur when they are close together. Now we see from the B image. This is the short section of the double helix field side, and this is from this image we can see the chemical structure of thymine, adenine, cytosine, and gynin. From this case, now we see for this structure. From this strain, we can see that the nucleotides are linked together covalently by phosphodiesters bond through the triaxine hydroxyl group of one sugars, and then 5-hexanes phosphate of the next. Thus, from this image, we can see also that each polynucleotide strain has a chemical polarity. There is two ends here. We can see this one for two ends, 3-hexane N and 5-hexane N are chemically different, where we can say that the 3-hexane N carries an N-link hydroxyl group to the 3-hexane position on the sugar ring, and for the 5-hexanes N carries a free phosphate group attached to the position on the sugar ring. Okay, I think this is the explanation about the structure of the DNA. Now we move to the function of the DNA. We know that the DNA has a crucial role as a genetic material in most living organisms. It carries genetic information from cell to cell and from generation to generation. Thus, its major function of DNA include replication, gene expression like storing genetic information, determining genetic coding, and then mutation, transcription, translation like a create protein, directing protein synthesis, and directly responsible for metabolic activities, evolution, heredity, and differentiation. I think it's enough.
Thank you so much, Ms. Alfiatur. So let's start with positives in your demonstration. So first of all, your mastery of the subject matter is very clear. You were able to discuss and hit the goals for this particular topic. Most especially, I appreciate that you took the time to discuss each of the parts of the DNA. For example, you took the time to explain how the nitrogenous bases are bonded together using hydrogen bonds, and that the phosphate and sugar backbone are bonded through covalent bonds, etc., etc. So that's very good. And also your structure and the way you presented the lesson is very simple. You were able to make a complex topic simple in the eyes of students. You used simple words which are very much understandable by, yes, which is very good. Okay. So anyway, it's only minor improvements. So maybe you start the lesson with a connection to real life, which is you did, right? Because you told them that DNA is the one responsible for the color of their eyes, of their hair, and that's also very good because students will be able to think that, okay, our lesson today is not just in the textbook, but it's also applicable in real life. So next time you can also share to the students the objectives for the day, so they will know what are they supposed to learn and what are their goals for the day, and if they are able or not able to reach those goals as well. But overall, it's very good demonstration. Thank you so much.
Okay, thank
Congratulations,
you so much.
Miss.
Okay.
So what's going to happen next is I will be waiting for the recording of this demo. I will pass
Okay.
the recording to our academic coordinator. And then after she has viewed your demo, we will contact you again for the next phase of the employment process. Okay, Miss?
Okay, thank you.
You're welcome, Miss. And so I will wait for the recording, okay?
Okay.
Thank you so much, Miss.
Okay,
God
thank
bless
you.
you. I hope to see you
God bless
again.
you. See you. Bye
Okay,
bye. Thank you.
bye-bye. You're welcome.